बिस्मिल्लाहिर्रहमानिर्रहीम। तो आज का जो हमारा टॉपिक है, पिछले लेक्चर में हमने क्रोमेटोग्राफी को स्टार्ट किया था. सो सम ऑफ द बेसिक्स ऑफ द क्रोमेटोग्राफी. सो टुडे विल स्टार्ट सम मोर बेसिक टर्मिनोलॉजी इन द क्रोमेटोग्राफी. तो क्रोमेटोग्राफी से हमने कहा था इट्स बेसिकली एन एनालिटिकल मेथडोलॉजी by virtue of which we can separate a given component mixture. It can be a mixture of cationous, या mixture of anions, or it can be neutral compounds. Now the first term जो है हमें यूज करेंगे यहाँ पे, that is कि इंस्ट्रूमेंट जो हम यूज करते हैं क्रोमेटोग्राफी में, इंस्ट्रूमेंट जो हम यूज करते हैं, जो मशीन हम यूज करते हैं in the broader term in the chromatographic analysis. Now that instrument is called as chromatograph. ये बहुत important है. So the instrument used, it's called a chromatograph. Second point, अब instrument जो है, what this instrument gives us actually, जैसे हमने कहा था absorption spectroscopy में, it gives us absorbance versus wavelength. And in the UE, this IR spectroscopy, our graph is इट्स लाइक ट्रांसमिटेंस वर्सेस वेव नंबर. तो यहाँ पे जो सिग्नल जो हमें ग्राफ मिलता है, सो दैट इज एक्चुअली द ग्राफ विच विल ऑब्टेन फ्रॉम द ग्राफ और डेटा. So it's basically data in the form of graph which is obtained from the instrument. So that's basically called as the chromatogram. So that is called as chromatogram. तो ये बहुत ही important. It's called chromatogram. Chromatograph is the instrument name. Chromatogram is the data we'll get, the graph which we'll get from the instrument through a PC, a computer. It's called chromatogram. तो chromatogram basically ऐसा होता है, typical chromatogram. तो इसमें फर्क होता है because we have different types of chromatography techniques. But in general, we have x-axis and y-axis. So along the y-axis, we have a term called as, that is the detector signal. And along the x-axis, we have the time. It's called as retention time, fine? Seconds. Detector signal. Now let us say we'll make a graph. Let us say this is a typical chromatogram. So it has some, these are called peaks. So this signal, it depends on which type of detectors you are using. Fine. Now, इसको एक rough image mind में create करना है ऐसे. So basically we have column is important component of all the chromatographies. This is a column. So हमने कहा था it's packed with some material. Now then, this column is packed with some material. It's called as the stationary phase. So the phase is basically which stays inside the column, and that phase which stays inside the column, that's called as, so the phase that stays inside the column, that's called as stationary phase. That's called as stationary phase. So that phase which stays inside the column, it's called a stationary phase. So we can put here different type of materials. For example, it can be a gel type material, like we do in the column chromatography. It can be a gel based packed material. It can be a highly viscous material that then gets we can pack this with a highly viscous coated material, and that actually gets coated inside the column, for example in the gas chromatography. So stationary phase, that way that actually stays inside the column, number one. So this is one more terminology. Likely there's one more phase that actually flows across the solvent, across this column, that flows through this or over this stationary phase. That's called as mobile phase. That can be mobile phase. So mobile phase can be a liquid. That can be your gas also, for example, in the gas chromatography. Fine, so it's like fluid. So this is the basic. So stationary phase and the mobile phase. Up now, the sample which we have to separate. For example, there are two components or three components. They can be mixture of cations, mixture of anions. They can be both. They can be neutral compounds. Now, when we have not separated those components, so we say now this in terminology we say that these are the analytes. So this is the analyte. So by analyte I mean to say that it's actually those entities or chemical compounds which actually we need to separate. So they're called analytes or sample. Now we will pour our sample or analyte over the column, and then with the help of mobile phase we are in a position to separate them. So now the solvent which comes from here, let us say one analyte comes. Okay. Now this analyte we can detect with the help of a detector. So we have a detector here. So I'm trying to draw a rough picture so that it will be easy for us. So now that detector can be, it is of different types. This detector can detect this with the help based on the absorption response. That analyte has some absorption value at different wavelengths. It can be on the absorption response, or it can be the conductivity based. So when the analyte comes out, now here it has some conductivity values. When the next analyte comes, it has different conductivity. So based on that conductivity, we can detect the analytes, likely based on the absorbance value. So now here, this signal can be either absorbance based, it can be conductivity. Usually, the signal it's electrical conductivity based. It's a conductivity that becomes, so usually it is conductivity based. It's usually. So your data or graph that comes from the instrument, that's called a chromatogram. It is basically sample, it's the signal response, detector signal versus the time. So these are the peaks. So each peak corresponds to a typical chemical species or analyte which was present in our crude sample, which is present in a sample from where we have to actually separate these. So these are the signals. Now, for example, this signal comes at this point. This signal comes at this point. This signal comes at, so there are different time periods. So we have one more term in the involved here, that is called as a retention time. Retention time in the chromatography. So retention time is basically how much time your analyte. So retention time is basically the time. So it's basically the time it takes for an analyte to come out of the column. So retention time is basically the time which is required to get the analyte out of the column. For example, हमने यहाँ से sample इसको डाला, फिर ऊपर से इसमें mobile phase is moving. अब उसमें जो analytes है, for example, for analyte 1 moves after 30 seconds. So it will take a lot of time to pass through this column, then undergo interaction, equilibrium between stationary phase and mobile phase. So total कितना time इसने ले लिया to come out of the column. Now that time is called retention time. So I can say retention time, it's actually the time which an analyte takes to come out of the column, or which a chemical species takes out to come out of the column. For example, यहाँ पे ये species number 1 ने, it has taken time from this to this. Let us say it's 5 seconds. So retention time is 5 seconds. Now this analyte, it has taken 10 seconds to come out. So its retention time is 10 seconds. This has taken 15 seconds. Just roughly. So it is analyte third has retention time of 15 seconds. So retention time is basically the time which a chemical species takes to come out from the column. That's called as the retention time. Now, इसी तरह से और एक term है इसमें involved, that is retention volume. retention volume. So we have one more term that's called as a retention volume. So retention volume is basically, is the retention time हमने कहा, the amount, the time a particular analyte it takes to come out from the column. That means once it comes to the column, detector will detect that. Or I can say the amount of time a particular analyte takes to up to which we will receive this maximum signal response. Same is the case in the retention volume. The volume of solvent, the volume of mobile phase we require. Since हम लोग इसमें मोबाइल फेज कंटिन्यूसली ऐड करते हो रहे हैं, the continuously flows through. So कितना volume of mobile phase required to make the analyte to come out of the solvent. Now that's called a retention volume. So retention volume is the volume of this mobile solvent, mobile phase required to make the chemical species, to make a particular component to come out of the column. So that's called a retention volume. अब retention volume को explain करने के लिए थोड़ा सा we'll go towards one more entity. We'll explain eluate, eluent, and there's another term that is eluent, and then another term called as the eluate. Technically, so eluent and eluate. Eluent is the fluid entering the column. हमने कहा था यहाँ पे ये stationary phase packed. Then हमारे पास एक mobile phase होता है, usually that solvent or gas. अब ये जो mobile phase which is actually coming out, which is a solvent, so that fluid which is entering the column, so that's called as the eluent. So eluent is basically the fluid that's entering the column, or mobile phase fluid. That can be gas or liquid. So it is the fluid that's basically entering the column. And since मैंने कहा कि यहाँ पे हमारे पास speed बहुत कम होता है यहाँ से, तो solvent is continuously flowing through and then it comes out. Now the fluid that comes out, so the fluid that is entering, that's called as eluent, and the fluid that's actually leaving the column, so the fluid that's basically leaving the column, that's called as the eluate. Now, if you go to the retention volume, it is the volume of the liquid. So, retention volume, it is the volume of the liquid that basically passes through the column while eluting a particular chemical compound. For example, हमें 5 मिनट लग गए एक component को निकालने के लिए. So, we can define the retention volume for this particular eluent. So, the amount of solvent or the amount of eluent that is required for a particular chemical component, that is actually called as the retention volume for that particular component. So that means retention volume is for different components. किसी को ज़्यादा लग जाएगा जिसकी interaction जो column में ज़्यादा time stay करेगा, that means obviously you require more amount of solvent, more amount of eluent to make this component to come out. So that means it has more retention volume. That means that has more retention time. So this is another terminology, retention volume, retention time. Then there's one more terminology. ना ये बहुत important है, that is the dead volume. Dead volume. Retention volume हमने कहा था कि the amount of eluent it will takes a particular chemical species or a particular analyte will take to come out from the column to get separated. So that much volume which is required to make this component to come out, that's called as retention volume. Now is दौरान when the mobile phase is passing through the column and the analyte is moving slowly, it's moving slowly and steadily through different equilibriums inside the column up to the point that it has not come out of the column. तो जब तक आपका एलिमेंट कॉलम, जब तक आपका एनालाइट कॉलम में ही रहेगा एट वायर आवर पॉइंट, तब तक जितना सॉल्वेंट रिक्वायर, दैट अमाउंट ऑफ सॉल्वेंट इज रिक्वायर्ड ड्यूरिंग passage to the column when the analyte was inside the column, that's called dead volume. So dead volume is basically the volume of the eluent. So it is the volume of the mobile phase or volume of the eluent. It is the volume of the eluent that's passed through the column. So eluent that's passed through the column while the component was inside the column. So while a component was inside the column, जब तक that chemical component और chemical species column में ही रहेगा, चाहे वो यहाँ पे हो, यहाँ पे हो, यहाँ पे हो, यहाँ तक हो. When it is inside the column, तब तक जितना solvent required, जितना mobile phase is required, that's called as the dead volume. Once it comes out, so जो सारा कुछ required solvent, right from this point up to the coming out of the eluent, that's called as the retention volume. So when the eluent was inside the column, that's called as a dead volume. Fine. So this is the concept of retention volume and the dead volume. Now this process of adding the solvent, process of then getting the eluents out of the column, and meanwhile getting this component separated. Now this process of this is called as the elution. So there's the term called elution. So this process is called elution. So elution means you are adding up mobile phase, adding up chemical sample, and then coming out from the column. This whole process, this is called as the elution. So this is called as elution. So these are the basic terminologies which we use when we actually discuss the chromatography. So these are the basic terminology which we use when we discuss the chromatography. Now uske baad we have some more terminologies. For example, those terminologies are related to the now I will go back to the column again because it becomes easy to understand. I will take the example. Just put image in your mind that there's a column which is packed with some material and there's a solvent is flowing through, or a gas is flowing through. So that becomes as a mobile phase, and this packed material, this column, it becomes this stationary phase. Fine. Now based upon this, what type of stationary phase or what type of mobile phase is used, we have the classification of chromatography. Classification of chromatography. यहाँ पे मैं दोबारा कहना चाहता हूँ कि I'm not going into the detail. I'm not going into the detail of the types of chromatography, but basic terminology यहाँ पे मैं understand करने की कोशिश कर रहा हूँ. So depending on which type of stationary phase you use, which type of mobile phase we're actually using, whether liquid or gas. So there are different. There's a classification of the chromatography. There are different types of chromatography. Type 1, that is the partition chromatography. Partition chromatography. And the broader sense, the second one, that is adsorption chromatography. So there are partition chromatography in the broader sense and adsorption chromatography. So partition chromatography में क्या होता है? याद रहे, again we have to keep in mind that we have a column packed with a stationary phase, and then we have और एक जो है phase that is mobile, which is moving, that can be liquid or gas. Partition chromatography में जो आपका जो liquid है, आपका जो stationary phase होता है, that stationary phase can be a solid, ये बहुत important है, or that can be a liquid. ये हम later भी understand करेंगे when we go to the different types of chromatography. So your stationary phase can be a solid or a liquid. That means जो packed column है, that can be either a liquid based material or that can be a solid based material. That can be a gel type, semi solid. ठीक. और जो आपका mobile phase होगा partition chromatography में जो mobile phase है, that can be liquid or a gas. Fine. इसी तरह से जो adsorption chromatography है, in adsorption chromatography जैसे phenomena भी बोल रहा है कि there is some sort of adsorption. Our stationary phase is obviously adsorption के लिए आपका stationary phase is solid. That is solid. आपका मोबाइल फेज, और मोबाइल फेज कैन बी liquid or a gas. Fine. यहाँ पे absorption बहुत easy remember इसके लिए क्या absorption that means solid phase surface. The material is getting absorbed on the surface. So it is stationary phase solid, mobile phase can be liquid and gas. Now if you take the example, solid example. Now as far as the partition chromatography is concerned, our example जो हम lab में use करते हैं, that is the paper chromatography. So paper chromatography, अभी मैं इसको यहाँ पे explain नहीं करूँगा. Just put on an example, paper chromatography. This is an example of partition chromatography. इस तरह से जो अक्सर हम कॉलम लैब में जो chromatography यूज करते हैं at UG level, PG level in the research, that's called as the thin layer chromatography, TLC. Thin layer chromatography. Thin layer chromatography. This is an example of partition chromatography. इस तरह से जो अक्सर हम nowadays जो बड़ी common हो चुकी है advanced level पे, high performance liquid chromatography, HPLC. खासकर कैलालिटी में बड़ी ज़बरदस्त एप्लीकेशन है इसकी, हाई परफॉर्मेंस लिक्विड क्रोमेटोग्राफी. सो दे आर द बेसिक मेन एग्जाम्पल्स एग्जाम में जो आ सकते हैं ऑफ द पार्टीशन क्रोमेटोग्राफी. लाइकली वी हैव द एब्जॉर्प्शन क्रोमेटोग्राफी. तो एब्जॉर्प्शन क्रोमेटोग्राफी के लिए आप सबसे बेस्ट एग्जाम्पल ऐसे यूज कर सकते हो, दैट इज द गैस क्रोमेटोग्राफी, GC. गैस क्रोमेटोग्राफी एक टेक्निक है. इस तरह से यू कैन यूज द कॉलम क्रोमेटोग्राफी. कॉलम क्रोमेटोग्राफी. So these are the just two important examples of the absorption chromatography. So this is the basic terminology used. We have some more terminology that will come in the third lecture. So today with the second basics or the basic terminology of the chromatography. Till then, stay